We have about 1 minute before we're starting, so if you could start to take your seats, please. Oh, good. Jason's back. Did you talk to him? All right, it is 10:45, so let's get this last session kicked off. Good morning. My name is Colleen Brust. I'm here from New Jersey. I work for the Department of Environmental Protection and the Marine Resources Administration, and I am just delighted to be here to the bitter end of this meeting, and bitter because it's been lovely, and so many thanks to the organizers for this. So this session is new technology. So I'm just going to call it Sexy Science Friday. And we are going to kick it off with a presentation from Katherine Gallagher called Using Autonomous Gliders to Understand Patterns and Drivers of Habitat Use for Baleen Whales in the New York Bight. So as Katherine comes up here, I would just like to remind you, please use the app. It's lots of fun. You can start putting in questions right now. And as questions come to mind, go ahead and type them in the app. So thank you, Katherine. Take it away.
Just close this out. Let's make sure Microsoft works with us today. All right. Sweet. Microsoft's working. Well, good morning, everyone. Thank you for sticking around till Friday, and thank you to Colleen for that introduction. As Colleen said, my name's Dr. Katherine Gallagher, and I'm a senior postdoc in Leslie Thorne's lab here at Stony Brook University. And on behalf of the team, I'm really excited to share our work today using autonomous gliders to understand where baleen whales are in the New York Bight. So baleen whale distributions on the New York Bight are historically poorly understood, although we are getting better and better each and every day. And this becomes especially important as those distributions of whales shift. Since about 2011, we've been seeing baleen whales like this guy right here, very close to shore along the coast of Long Island and New Jersey. And as we might be aware, the North Atlantic right whale distributions, they are using more and more of southern New England. So the New York Bight could potentially be a future habitat for them. So we really want to understand how whales are using this space. And as many people have talked about before, the Port of New York and New Jersey is one of the busiest ports, if not the busiest port, along the East Coast. So there's a lot of activity in this area that we need to be aware of. And the reason we are all here today is the wind farm development in this region. So before we potentially take valuable habitat from these whales, we first need to understand what habitats they're using. I won't beat a dead horse here. We've talked a lot about how we monitor whales and other animals here all week, but just as a quick recap, there are a lot of different ways we can do this. So a lot of this work has been done visually, either through aerial surveys or boat-based surveys like we do in the Thorne lab. These techniques are great because they allow us to actually count the number of individuals that are present, but they do take a lot of resources to conduct. You need a lot of money to go out on boats. You need a lot of people in and on those boats and in these planes to go out and do this work. A lot of us are familiar with a relatively not super new technology of passive acoustic monitoring, so this field has really exploded in the past few years. There's a lot of passive acoustics that can be done off towed arrays, off of vessels, but really what's taken off in the past few years is passive acoustic monitoring on moored devices as well as on autonomous vehicles. And so that's what I'm going to be talking about today. We're going to be focusing specifically on autonomous gliders, which again are not a new technology, but the combination of these two technologies has really taken off in the past few years. If you're not familiar with how a glider works, gliders are buoyancy-driven instruments that move in the water column by changing their buoyancy. That allows them to fly in this sawtooth pattern here. They do it a little bit more smoothly than PowerPoint allowed me to animate. But all this time, they are collecting a lot of oceanographic data, and in our case, they are also listening for the calls of baleen whales. Occasionally, these instruments will come to the surface, where they can communicate with us via a satellite phone in the tail. They transmit data and information to us here on shore at Stony Brook and other places like Rutgers. And this surfacing opportunity allows us to send new information back to the instrument before it continues on its way. So our questions here are quite simple. We're basically interested in what species we're hearing and when and where we're hearing them. And notice, and just keep in the back of the mind that we're looking at where we're hearing these species, not when we're seeing them. As many of you are probably aware, passive acoustics really just relies on us listening to the animals. We don't know exactly where they are in this space when we hear them or how far away they are, although we can probably make some educated guesses. So just, again, keep that in mind as we get into the results in a little bit. Our study area here is the New York Bight. This is where we are sending our gliders. And you'll notice all of the different wind lease areas on this map. So how many gliders have we thrown in the water? Right now I'm going to be presenting data from 21 different glider missions spanning from 2020 up until this February, but I'll also mention that one glider just went in the water about 20 minutes ago. So we do have a few more missions, more recent missions that I'm not considering here. All of these glider missions are equipped with a digital acoustic monitoring or DMON instrument. This is the instrument developed out of Mark Baumgartner's group at Woods Hole. And we are using his low-frequency detection classification system, or the LFDCS, to detect humpback, say, North Atlantic right whales, as well as fin whales. We currently have over 500 deployment days and 10,000 kilometers surveyed. And I want to give a quick shout out to NYSERDA here. A portion of these missions, as well as my time, are funded by a NYSERDA project that specifically targets monitoring in the wind lease areas. And on top of, we've had about the 6th glider mission just went in the water. And I'm also leveraging a lot of other projects and other previous glider missions by the Stony Brook team and also the Rutgers teams that have these sensors on them to get to this 21 glider missions we have. This map here on the right illustrates all of our glider missions to date that I'm including in this analysis, and the ends here just illustrate the number of deployment days, the number of days we have a glider on the water in each of our study years. So you can see when our monitoring program picked off with NYSERDA in 2023, significantly increased our survey effort. So starting to look at what species we're hearing at what times of year. The x-axis here is time in months, and our y-axis is the proportion of days that we had a glider in the water and what proportion of those days we determined a whale to be present. Starting with our humpback whales, we have a very strong pulse of whales in the springtime, and we attribute that to the spring migration of humpback whales coming up from the Caribbean and migrating to their foraging grounds here in the New York Bight, as well as in the Gulf of Maine and other places in higher latitudes. For sei whales are one of the endangered species that we hear in our acoustic calls. So they're not as present as the humpback whales are, and there's another similar peak in the springtime that could be a similar spring migratory pulse. Our North Atlantic right whales appear to be overwintering in this area. Again, detection rates are pretty low. Given the population size, that's not a huge surprise. But we do hear them more often from the fall and into the following spring. And we also see similar patterns for our fin whales. We see them most often in the late summer in the fall as well as the early spring. So now thinking about where we hear our humpback whales, this 4-panel plot here illustrates 4 different seasons. We have winter in the top left, moving into spring, summer, and fall. These are the same maps on all 4 panels with the wind lease areas in the lighter gray in the back. The black lines here are the glider tracks, so you can see where the gliders were when it detected these whales. And all of these colored points are when we heard a whale detected, when we had a confirmed whale detection. So the whale, again, is not precisely in this location, as far as we know, but it is in this general vicinity. Starting again with our humpback whales, we really hear the humpback whales shelf-wide, especially in the winter and spring. And we also see them in some of the wind lease areas like Empire Wind and Blue Point Wind. I'll point out also that we primarily really only hear humpback whales south of the Hudson River Valley in the summer and fall. We need more survey effort on the Long Island side of the Hudson River Valley to really confirm this, but there could be some interesting sidedness to this environment. Sei whales, again, we don't hear them as often as the other species, but we do hear them traditionally further offshore, although we do generally hear them closer to inshore on occasion. And they also have that association with the Hudson River Valley, especially in summertime. North Atlantic right whales, again, we hear them pretty much shelf-wide over the winter months. But we do hear them closer to shore generally in the fall. And then last but not least, our fin whales. We basically hear fin whales everywhere. No surprise there. But we do see them pretty much across the continental shelf almost all year round when we do hear them. So what does all this mean? We've demonstrated, and we're obviously not the first people to do this, that gliders can detect whales here in the New York Bight, and that they do give us a really good understanding of baleen whale presence in the New York Bight year-round. A lot of these results line up pretty well, actually, with the results that WCS showed earlier this week. So that's really great to see that we're seeing similar results across different platforms. We saw known ecological processes, such as the spring migration of humpbacks. We saw that North Atlantic right whales appear to be overwintering here. And again, that possible association with the Hudson River Valley, although we need more data to confirm this. Gliders we've also shown are a really good platform, and they really span a nice sort of intermediate between stationary platforms that give us a really long time series and mobile boat-based surveys that give us a lot of spatial distribution, but really only occur once or twice a year. Across all 5 of our years, we essentially have a year-round monitoring of this area, so they really provide that near continuous monitoring, which is really helpful and really nice to see. But again, we do fall to some of the downfalls of just general PAM, passive acoustic monitoring in general, and we are reliant on the whales to call for us to be able to detect them. So we have a lot of future work ahead of us, as well as future opportunities to improve, including looking more inshore. We obviously see a lot of whales inshore, but we are not detecting them, either because our gliders are too far offshore or because they're not calling. And we are really excited to continue to build this multi-year time series. I also did not mention at all of the oceanographic data that gliders are able to collect. So using all of this different data, we're hoping to be able to build models to define preferred whale habitats based on the oceanographic conditions that the gliders are measuring. So with that, I thank you all for sticking around to Friday again and for your attention. I want to quickly, again, give a shout out to NYSERDA for funding both my time as well as the monitoring project. The New York DEC also covers and funds another additional set of glider missions, as well as previous missions funded by New Jersey RMI and Ocean Wind. And I look forward to your questions. Thank you.
Thank you so much, Catherine. I hope you're all as inspired as I am by that presentation and seeing the tracks of the animals out there that we're all working hard to protect. So thank you. Next, I'd like to invite Jason Adolf up, who's going to give us a talk on the integration of non-extractive environmental DNA in monitoring impacts of offshore wind development. And again, write your questions down in the app.
Thank you, Colleen. I'm going to try something here with a pointer I learned from being a teacher. All right. So I'm happy to be here on the last day. I'm happy you're all here still on the last day. I want to start by acknowledging my co-authors. Keith Dunton is in the audience right there. Shannon O'Leary is a molecular ecologist that we work with from St. Anselm. Sam Chu Chin was with us. He's left for NOAA, had a baby, started a family on the West Coast. And Kieran, Nick, and Aaron are right back there. They are our field/lab technicians. None of this would happen without this team. So we're here because of offshore wind development. Don't need to go through this in detail with this group here, of course, but it's worth saying that we're doing this to mitigate a climate crisis. And that's where all these energy goals come from and all this planned activity comes from. But the questions we need to ask and that we ask are what might this do to the biological and chemical conditions on the shelf? So we've formed these fisheries monitoring plans, of course, which essentially ask, from my perspective, what fish are there and how might they be affected by development? There are lots of effects that have been gone through. The reef effects, electromagnetic effects of transmission lines that could change distributions, and also oceanographic conditions that might change and have an impact on fish community composition. We're also focused very strongly, I think it's important to bring up, on how we measure things now in order to detect a change 6-10 years down the line. And it's all very complex. What I'm going to talk about today is environmental DNA and genomics approaches to fisheries and how that can help with these things. So what is eDNA? eDNA is DNA that's collected separate from the organism that produced it. In this case, fish shed DNA in mucus, body fluids, tissue, excrement, and we can collect that. We collect a liter of water from either the surface or the bottom of the ocean when we're collecting eDNA and filter it onto a filter, and then extract that down to a purified DNA extract. From the purified DNA extract, there are two basic routes of analyses that people go through. One is a PCR, qPCR, ddPCR route, where you're using probes to look at single species at a time. And there's a metabarcoding route, which I'm going to talk about today, where you use so-called degenerate primers to amplify the DNA of a group, such as fish, teleost, elasmobranchs, or different sorts of vertebrates. And from that, you get community data. So we can ask different sorts of questions from these different sorts of approaches, presence-based questions, I'll call them, things like what's there, how does what's there compare with capture methods, and models like species occupancy models can be done. And then there's community-based metrics that can be done from the community data where you're considering the community as a whole, and this is very powerful and very amenable to eDNA sorts of data. And then finally, for the nerds out there who care, it does matter how you do this stuff, not only in QA/QC, but also interoperability between people doing eDNA. So I'm going to put that slide up there. I'm not going to go through the details. eDNA is having a moment as well. Back in April, I attended a congressional briefing with a bunch of people from Cornell and MTS, where we were telling our congressional and senatorial staff members about the upcoming National Aquatic eDNA Strategy. Hands up, how many people knew we had a National Aquatic eDNA Strategy? How many of you know now? Right on. So grab it at the QR code, read it. This comes out of the White House, and this is encouraging federal agencies to further adopt this technology, which is ready to go, but currently is housed in research laboratories and not really ready for wide-scale use, or not adapted for wide-scale use through things like standardization of protocols and data. And within that national policy, I'll say that renewable energy, offshore wind, and NEPA compliance was called out as a specific case use for environmental DNA and genomics approaches to fisheries. Our group at Monmouth University has been doing eDNA in an offshore wind context since 2021. The sites shown here are all the sites where we have samples. We work with, we started down here with Rutgers University in the site formerly known as Ocean Wind 1. We work with Inspire Environmental in the Empire site up here, pairing with Brust surveys. And the rest of the dots there are what I'm going to go through in more detail, our RMI project with the New Jersey RMI program, which includes 7 different sampling campaigns. So the blue dots here is a grid of unpaired eDNA sampling that we do around, because they're active at the time, Atlantic shores and Ocean Wind 1 sampling areas covering inshore to offshore, north and south of the wind areas. And we also sample along the shoreline in the surf zone where you can't do community sampling very well otherwise. We also included a community sampling component in here where we train and equip, Aaron leads this, community scientists to sample eDNA, and we look at how well their data comes out compared to our data. We're pairing with 3 ongoing capture surveys run by the DEP, the Ocean Trawl, which takes place here, the Raritan Inventory Project up here, and the Artificial Reef Survey at Seagirt and Little Egg. And then, of course, we're pairing eDNA samples with Dutton's acoustic telemetry terrain, which are spread around the entire shelf, I'll say. We also collect environmental data with all these samples because we understand that teasing out a wind farm effect from other effects will require incorporating environmental variability. So contact me if you want to talk more about the data. I'm going to go through a little bit data quickly. The plot you're looking at here is species richness, how many taxa were found in a sample. There's 5 different sampling bouts or trawl surveys. The green is the eDNA samples, the orange is the trawl samples, and we see a pattern that we've seen in other studies and other paired studies. We tend to find more stuff per sample in the eDNA than in the trawl. Another way of looking at that, I did a little modeling exercise, is the cumulative richness as you consider all the samples taken in the survey. The orange line are the trawl samples, and the orange dashed line is the maximum cumulative richness found in those samples, and the green is the eDNA. And you can see that eDNA cumulative richness exceeds trawl richness with far fewer samples, and it just illustrates the utility as a biodiversity tool of eDNA over methods like trawling for that one purpose. Now, richness means that there are different taxa being found, so I tried to summarize that here with these plots. On the left, we're looking at as a proportion of samples taken, all surveys combined, taxa that were found in the eDNA without a trawl match. And you can see there's a high proportion of samples where they're found, and this partly explains the difference in richness I just showed you. If you look at the taxa, too, you'll see that there are some things that probably aren't surprising, like little bay anchovy and strong swimmers like sturgeon and structure-associated stuff like tautog. DNA is a dispersed signal. If there's a fish over there, its DNA is probably everywhere within a kilometer. You might not catch it with a trawl, but you're going to catch it with a liter of water, more likely. On the other side is the bad side. These are taxa that we found in the trawl without an eDNA match. And the first thing I'll point out is that except for winter skate or little skate, which I don't have a good explanation for, the proportion of samples in which that happens is very small compared to the other graph. The bars in red we don't catch in the eDNA because we don't have their reference sequence in our database. Now, I was talking to Mats at this meeting, and he pointed out that this particular one, Blue Runner, is probably corresponding to we have a giant trevally sequence in our reference database. I just haven't corrected it yet, and I had submitted the slides already. So there's lots of reasons why a taxon would end up on either of these graphs. And And it's something that we're very interested in understanding the gear bias of different techniques and what we catch and what we don't catch in each method. Metabarcoding is not the solution to determining absolute abundance of fish, the way a trawl does fish per meter square or volume of ocean, but it can return relative abundance, that is to say, how relative to all the fish in an assemblage, how dominant or not dominant is it? And here, what I'm showing for the Ocean Wind Survey, which we did in collaboration with Rutgers, 2 trawl surveys, and the 5 trawl surveys with DEP maps over here. On the x-axis of both of these is the relative reed abundance for a taxon. Each different colored dot is a taxon. The triangles are sharks and rays, elasmobranchs. And this is the relative numerical abundance from the trawl catch. And you can see that they do line up in a significant relationship, and that's good. So concordance, biodiversity is a very strong suit of eDNA. I want to move on and show very quickly what we're doing with community analysis. So these are techniques that you're probably familiar with. Ordination techniques where you get your data and you ordinate it like this and separate it in variable space. What I'm going to show you is we've been applying a k-means machine learning algorithm to pull out similar compositions from the eDNA data and identify different community compositions of fish. The way I present this is that when you look at these maps, every color represents a statistically different fish community composition as determined by k-means clustering. To figure out what fish are in that cluster, you come over to this map and you just scale up the cluster. It's too much to go through here. I've already got the 2-minute warning. But I do want to highlight one thing. Part of our sampling, as I mentioned, is the artificial reefs of New Jersey. And you'll see that the shapefiles, the 2 red shapes there, and you'll see that there's a different color symbol over those. And that corresponds to clusters or compositions that are dominated by what you would expect, conger eel, black sea bass, and tautog, different from the surrounding zones. So this sampling of the artificial reefs has given us a good field test of the resolution capabilities of water column eDNA sampling for determining local fish community composition. So I had AI draw this metabarcoding picture. But in summary, we're using eDNA as part of fisheries monitoring plans. We're focusing on establishing an ecological and a methodological baseline for the technique, pairing with capture methods, and the strengths that we're finding are in alpha diversity and beta diversity, and there's so much more to do with this. I didn't mention, we also find marine mammals, bird genes, and turtle genes in our samples with the fish. I just wanted to throw that up there. Find your favorite animal on the left there and call me. In bigger conclusion, eDNA is really a new set of tools. It has great methodological resilience. It's very simple to do. You can do it in places you can't do other sorts of fisheries techniques in ways. And in the future, one of the most important things is going to be to put this on AUVs and collect things autonomously. I got the stop sign. I have to. Thank you very much.
Thank you so much, Jason. Next, help me welcome Kyle Landolt, who's going to be talking to us about machine learning and high-performance computing for the detection and classification of marine wildlife in digital aerial imagery. Keep those questions coming.
Hey everyone, thanks for being here for Sexy Science Friday. I'm here today to talk about some work we're doing to use machine learning and high-performance computing for the detection and classification of marine wildlife in digital aerial imagery. And unfortunately, I don't have any cool AI pics of metabarcoding, kind of horrifying scanners going on. Next presentation, though, I definitely will include some. So this is a multi-agency collaboration. I'm with USGS, and we work with BOEM, the U.S. Fish and Wildlife Service, the University of Michigan, the New Jersey Audubon, and Quanterra, a private contractor. And the main goal of our project is that we want to use machine learning to count wildlife targets in digital aerial imagery. And to do that, we focus on 5 main components. For this talk, I'll mostly touch on the work we're doing for machine learning in a HPC environment and talk about some of the work we're doing for data management and generating products that are useful for decision makers. So we're transitioning into these remote sensing and AI surveys because it allows for increased safety of crew members if they're able to fly at higher elevations. And it also allows for the digital record of targets to be collected so we can do analyses further down the line as models improve and we can generate more accurate statistics. And to do that, we're working with the U.S. Fish and Wildlife Service who are developing the Seabird platform, the Safe, Efficient Aerial Bird Detection Platform, which is one of the better government acronyms I've heard of. And here we have Mark Conniff with Fish and Wildlife Service showing off his plane and the hardware that's in the plane. So it's a 7-camera sensor array that's been optimized for very high quality, high resolution imagery. And there's also some in-flight machine learning capabilities embedded in this hardware. We work with other image data sources delivered to us by like APEM and Normando, other prior U.S. Fish and Wildlife Service missions like on a phase 1 platform. But the general consensus is that we work with just 3-band imagery. The pixel sizes often range from 1 to 1.5 centimeters, even sub-centimeter a lot of the time as we do more missions. And for machine learning, we downsample the imagery to 8-bit JPEGs. So how do we handle machine learning? We break it down into 2 main components, the first being detection. So how do we detect wildlife targets in an image, and the second being classification. So once we detect an object, how do we get to it like a refined taxonomic ID for that particular individual? For detection, we made this benchmark detection data set. So taking parent imagery of varying sizes, we crop it down into uniform sized tiles of 1024 by 1024 pixels. In our current data set, we have around 46,000 tiles that we spread across a train test validation data set. Half of our tiles are just empty background water, so there's no wildlife in them, and half contain at least one wildlife object. And it's a very bird-heavy data set, so out of the 84,000 objects we have, around 65,000 of them are bird objects. We trained a YOLO V8X model using the Ultralytics YOLO V8 open source code base, so anyone can go in and download and use this software. And we trained it on the 8 course classes. So again, bird being the most common class, cartilaginous fish like rays being the second most common, all the way down to reptiles, or sea turtles being our most rarest class in this data set. And we trained this model using tallgrass. So it's the supercomputing environment that USGS owns and operates in Sioux Falls, South Dakota. We used 1 deep learning node to train our model, and it has 4 NVIDIA V100 GPUs, and it took around a day and a half of just pure training time. After training, we ran our model over the test data set, and we generated how well it performed for each class. And so we were pretty satisfied with our bird, cartilaginous fish, artificial, and reptile class performances, and not as satisfied, but generally still happy with the performance of the other classes. I will note, though, that this is like on our own unique data set for our test data set, and it might not translate well to other imagery if there's like increased glare or sea foam or algae that our model hasn't seen. You might expect the performance to drop a little bit. We can go a little bit deeper into the performance and looking at the normalized confusion matrix. So here we can plot like the truth along the predicted values to kind of see where the errors lie for each of the classes. And we can look at like when there are birds in an image, generally they're missed as background objects, so it doesn't do a great job there. Other labels though, like mammal, it might have a successful bounding box drawn around them, but mislabeled as like a bony fish or a cartilaginous fish, for example. And then in the top right corner, you can see that when there are background objects like sea foam or glare, sometimes it does call them bird objects. So as the U.S. Fish and Wildlife Service has been collecting imagery, we've been archiving them using these Black Pearl devices in Sioux Falls. And today we have around 2,800,000 images in our archive. and we can continuously receive hard drives of imagery, and so we're pushing more than 4 million images in the next month as we get another batch delivered to us. And the really nice thing about our imagery being archived at AROS here is that it's really close to our tallgrass supercomputer, so we can really quickly do some large-scale test runs. So about a month ago, I took 2 million images that span collection dates between 2021 and 2023, ran it through a model which took around 2 weeks across 12 to 24 GPUs, and we can enumerate the total detections of birds, for example, to be around 240,000 objects. On the left map here, you can see the total number of image footprints that test run occurred on, so it covers a lot of the East Coast, and we can zoom in a bit closer and look at these spatial and temporal sort of relationships of these bird predictions. We can zoom in to hotspots that the model is finding a lot of birds at and kind of visually verifying that these are true positive bird detections that the model was, we can assess that it was performing fairly well in this area. We can use it for other data sets too. So a GeoTIFF data set delivered to us by Normando and APEM, it's essentially the same thing. GeoTIFFs are a little bit larger, so they take a little bit longer to run through our model, but we can generate the same sort of statistics where we can run it through and just collect the output and enumerate the counts. The nice thing about GeoTIFFs, though, is that we can plot individual objects spatially. So each little red dot you see here is a single bird object, and with a heat map plotted about it, so we can get really local densities of wildlife objects. And we can crop out the GeoTIFFs with the bounding boxes and plot them in like a common GIS software and label them by the label that the model thought it was with the confidence value alongside it. So it's really nice to take a data set that was many terabytes in size. Like imagery is not a small thing to archive. Taking a data set that's many terabytes and condensing it down to a data set that might be many gigabytes in size makes the usability of this much more achievable. We can begin to use that common GIS software. I don't know if anyone here uses QGIS, so much better than ArcMap. We can zoom into these little dense flocks of objects and kind of confirm, hey, these northern gannets are being successfully detected. That's really good. Other black scoters are being successfully detected. Also really good. This lone bufflehead successfully detected. Also really good. And then other non-avian objects like sea turtles, whales, cartilaginous fish, also successfully detected. I chose the thinnest red line I could find for this presentation, but that is a successfully detected whale. But again, it doesn't perform well all the time. We get some dense patches of bird detections in this image. When you zoom in a bit closer, you can realize that it's a false positive, it's just a bunch of sea foam that the model thought were bird objects. But it is nice to kind of visualize this data spatially, and so a user can interpret the results and make better sense of it as they move along their own data sets. So the current workflow looks like this, is that we take these large image data sets, we pass it through a YOLO detection model, we get output detections over wildlife targets, and we can begin to pass those output detections to a refined classification model. Some current work we're doing is just starting back at the YOLO V8 model and expanding on those 8 course classes into much more refined taxonomic families. So for example, instead of just saying something's a bird, we can say some things like an auk or an alcid or a gull, or instead of just a cartilaginous fish, we can say that's a white shark or like a hammerhead shark family. Some work we published last year is using classification models with a taxonomic hierarchy embedded into it. So we can have, like, for example, 2 black scoter species that can be aggregated up to a scoter superclass paired alongside other non-scoter values like common eider, long-tailed duck, non-target species. And this really helps boost model classification performance beyond just treating everything as a flat list. So the taxonomic hierarchy does help us here. The current work we're doing with the University of Michigan is looking at semi-supervised classification using unsupervised selective labeling. So as I showed earlier, we have like 240,000 bird detections. We want to know what are the most representative and diverse objects in that data set so we can more quickly expand and navigate that data set to select data, pull it down for humans to review, and more quickly boost model performance faster, I'll say, than doing everything in that data set. So just selecting 12% of that data set, for example, would more quickly improve performance than doing all 240,000, which is some pretty interesting work. And the most important thing I'll mention here is that we find it's really critical to include the human in the loop. So we don't want to just trust AI for everything it says and make decisions off of that. We want to review performance of the model output using this AWS SageMaker ground truth tool and manually correct the data where the model wasn't performing well, and then pass those corrections to a refined model and retrain it, and then take that retrained model and reapply it to our imagery. So it's this nonstop cycle of correcting data, retraining the model, and reapplying the model. And lastly, we're taking that coarse detector and integrating it with a refined classifier. So instead of just detecting a bony fish, we can detect ocean sunfish. Or instead of detecting just a cartilaginous fish, we can detect like a scalloped hammerhead. And hey, that's critically endangered, so let's make sure we can pass that to a human for review and make sure we're not making decisions based on just what the AI says for these critically endangered species. That's all I have for you today, and thank you for your time.
Thanks, Kyle. I don't know if any of the rest of you here are getting this really awesome feeling of, well, a couple years ago, these are the projects that we were really hoping to see, and here we are in 2024, and people are doing all that amazing work. So next, I would like to invite up Sarah Courbis. Let's give Sarah a warm welcome.
Alrighty. Oh, we don't have the little pointer one. I'm not as savvy as the person who knew how to do it on the machine here, so. All right. Thank you so much for hanging in here toward the end. I'm going to talk about something a little different than the other speakers so far. I want to tell you a little bit about a project that we did in collaboration with Biodiversity Research Institute, looking at technology gaps for monitoring birds and marine mammals at offshore wind facilities. The National Offshore Wind Research and Development Consortium put out a call for proposals to develop new technologies to help with this type of monitoring. And Kate Williams and I had a conversation and thought, another really important thing to know is what are the gaps that scientists need in order to get statistically robust outcomes from these types of studies, and how can we integrate this into the operations and infrastructure of offshore wind. So we put in a proposal to also look at that in the context of new technologies, and we're happy to be funded by NORDC. I'd like to thank all the folks who assisted with this project. This isn't an exhaustive list, but we also had a advisory body, and then of course a large team that supported us through this work. So the goal here was to give recommendations to NORDC about targeted development of technology that was going to help to improve the statistical robustness of data for long-term and short-term monitoring, as well as integrate that type of technology into the direct infrastructure. We didn't limit ourselves to East Coast or fixed wind, so this was a little bit broader project that also included floating and a variety of geographies where wind is proposed in the United States. The first thing we did look at were priority research questions, but we didn't reinvent the wheel on that. There have been a lot of different workshops and groups that have gotten together and published materials on that. So we gathered that from prior work and a literature review, and then we also used some workshops to get at some of the other questions that we were asking. So we ran 3 workshops, one on marine mammals, one on birds, and then one specific to integration into infrastructure and operations. So trying to bring the folks who run vessels and the folks who actually develop the infrastructure together with some of the experts in the technologies to see how we could do a better job of integrating. We also did develop 2 databases just to give us some information about what technologies were actually available and what they could do, and what technology readiness level they were, and things like that. So if you're interested, we published all this on our website. That's NORDC's got it on a website. I have the website at the end of this talk, and then We also have a publication that's coming out soon in Press and Marine Technology journals. So you can get access to those databases on the website at NORDC. Those are static, but we did collaborate with RIWI and NREL and other folks who have ongoing updating databases, so it's all fairly complementary. So, as I said, we started with looking at the priority questions based on previous work, then went ahead and did these workshops, and then finally, adding in the databases, provided a final report to NORDC with the hope that they can use this information to help them do RFPs and target some of the money in development of technology to focus on some of these gaps. It also helps folks know what the gaps are so they can work toward them, right? So they have some idea of what it is that's needed. So when it came to the priorities that folks had identified around research questions, a lot of them focused on occurrence, the conditions and stimuli, responses to different types of stimuli, those long-term population consequences and potential cumulative effects. And then there, although not really entirely a technology question, although to some extent, the data standardization, access, sharing, all that kind of stuff that we all talk about I've seen talked about at many of these workshops here. The two databases that we put together have a lot of different technologies in them. Of course, technology is constantly changing, so those databases were finalized in August last year, I think, so they're already almost a year out of date. But they do give you an idea of what kinds of things are out there and what some of the constraints and opportunities are for using technologies. At the technology surge yesterday, there was a lot of talk about repurposing existing technologies, the fact that there are, in fact, many technologies at a readiness level that you can employ right now and maybe tweak or change in some way a little bit, as opposed to reinventing entirely new technologies. So it's definitely worth looking at. When it came to limitations on the technology, looking at these gaps, we were surprised to find that often marine mammal and bird technology limitations were pretty similar, actually. There are a few things that weren't, but generally speaking, there were issues with the data transmission, the data quality and resolution, integration of data streams, a lack of communication, and a timeline mismatch. And we talked a little bit about that at the tech surge yesterday. One of the main things which probably has been in the back of your mind anyway is the fact that there is a mismatch in time between the different schedules for producing a wind farm. So you have a financial schedule, you have an engineering schedule, you have a procurement schedule, and you have an environmental and permitting schedule, right? That environmental component where a monitoring plan may come to finalization or regulators may decide exactly what you need to monitor generally comes very late in the process. So a lot of the engineering and design and procurement have already happened. This makes it very difficult for developers to go back and retrofit things like a bird radar or what have you, or even in operations to say, well, we want you to do this monitoring that requires deploying an AUV. Well, we didn't procure a ship that we can do that with in our operations. We'd have to go out and do a completely separate procurement and a completely separate activity to do that. So if that mismatch in time is something that we think is really critical to try to address. And of course, the financial milestones are really important too, and I think sometimes we forget that in the sciences. Like, if we don't align the milestones they need to meet for financing, they'll lose their financing, right? So there's things that they need to do in procurement and in design that just don't, if they don't do it on time, they're not going to be financed. So it's really important that we move that environmental schedule back rather than thinking we're going to move the procurement schedule later in the process. Generally speaking, some of the constraints on the physical technology was looking at power supplies, the availability of space on platforms, issues with masking. If, for example, if there's too much noise, it's hard to use PAM. If you have radars, they can be blocked by infrastructure and structures, things like that. The stabilization, I think a few folks I've seen talk here have talked about how, particularly for floating platforms, they're moving, so if you're putting equipment on them, you have to be able to stabilize that in some way. Also, the access to the equipment. I've been in rooms with researchers, and this is a couple of years ago, but saying, oh, well, we'll just go on the platform and we'll maintain our equipment or stick our USB stick in there and get our data or whatever. These are not things you can really do on an offshore wind platform, right? This is a safety issue. There's a lot of health and safety considerations around how this equipment could be used. So we want things that are more autonomous, have redundancy, will last a long time, are very robust to the marine environment, and have hopefully adequate power supply and data transfer capabilities, that there really doesn't have to be a lot of access to those things. We talked about integrating technology into the actual platforms themselves, the infrastructure of the wind farms. Some of the things that we talked about with our workshop are listed here. I don't need to read through them per se, but we were looking at questions around how can we use the existing equipment in some cases, like if you have sensors, for example, on moorings that are meant to detect pressure and changes that tell you when something is attached to the mooring. That's the question we're asking as well in terms of potential for marine debris to get attached and affect wildlife. So can we make sure that we're purposing this in a way that collects data on both fronts? There's lots of cable that is used in this, that has fiber optics. There are opportunities for using sensors that are already within the cables or even adding sensors to the cables. So there are a lot of things we can do with the infrastructure, not just around putting radars and things up on the platforms, which probably a lot of people have talked about. So here's a list of some of the improvements that we felt were necessary in order to make sure the technology could be out there and collect the data that we needed. Some of these are pretty obvious. I think one thing that I didn't mention is that trying to be able to detect small-bodied birds and high-frequency cetaceans were two things that kind of came up from a biological perspective, as well as improving algorithms and things like were being talked about in the last talk. It's important to be able to integrate data sets. So at other talks, we've talked a lot about how we can get data more standardized and get it out and available for scientists to use in larger scale studies. That's going to be really important. Commercial scale production of things that we don't normally produce at commercial scale to is an issue. So, for example, marine mammal tags are often made in somebody's basement. This does not make for very, a lot of consistency or availability. And then one of the things that I wanted to make sure I emphasize here, and I'm almost out of time, but that integrating into the infrastructure, one thing that we talked about was there could be the potential to, in the early stages of design, try to design a space on turbines. Or if you're working on some other thing, I mean, nacelles and things are harder. But if you have a space that's dedicated for technology for wildlife monitoring, where there's power available, where data transfer is known, the size, weight, way it could fit in there is known, then technology can be designed to fit to that space. It's like you have a plug in the wall, you can stick a refrigerator in it, or you can stick a microwave in it. You can put anything in there that's doing what you want it to do, and you know what the limitations are. So in conclusion, we summarized some of the gaps and potential solutions in this report. I encourage you to take a look at it. A lot of the gaps are similar between birds and marine mammals, although birds have had a lot more work on land, obviously, that helps to feed into the technology for birds. We need to make sure that we're collecting data that are statistically sufficient. And so that's something that's really important that our technology be able to do. And then this can be used to prioritize funding for different types of technology. So thank you very much. Here's the website. If you, I know that's really long, but I think are these available somewhere? If the presentation's available, you can get it from there. If not, just go ahead and email me, and I'm happy to send you the link. Thanks very much.
Thank you so much. So I just checked with Morgan on how we can view these slides later, and the info I got is that they will be held in CVent for how long? 90 days. So you have 90 days. All right. Next up, we have Audrey Bard, who's going to be talking about detecting whales using infrared technology. So welcome, Audrey. Yes, of course. Okay, let's try that again. Welcome, Audrey.
Thank you. So good morning. My name is Audrey Bard. I'm with Equinor, and I will present on a case study of a trial that we did for a long-wave infrared camera on the Empire Wind project. And I'd like to acknowledge Toyon's work on this. Unfortunately, they couldn't be here today to present, but they did most of the work on this, and it is their equipment. So the Marine Observer is the name of this system here, produced by Toyon. So we deployed this technology on the Empire Wind surveys. It was deployed independently and alongside protected species observers who were there to support mitigation requirements. So it was really meant to be a technology demonstration trial. You can see here the area where it was trialed in the Empire Wind lease area, and the data that we have covered 25 days or 600 hours, and it was in August and September of 2023. There we go. So you can see here on the image the camera installation. So this particular configuration of the Marine Observer is called the WellCam 66. It has a field of view of 66 degrees, so it's 6 different lenses, and it includes internal electronic image stabilization to kind of counter the movement of the ship, and it has a frame rate of 30 images per second or 30 hertz. It was mounted on the deck above the bridge, and it was looking forward and slightly starboard of the vessel. You can see a little diagram down there to kind of show you what the installation was like, and you can see the interface here with the AI detection of whale blow. So this is what the either the system operator can see or PSOs potentially. So before looking at some results, I want to just align on some definitions of what the terminology that I'm using. So when it comes to the IR camera, what I'm calling a confirmed detection is an IR camera detection which has been confirmed by 2 independent human analysts looking at the footage as being a possible, likely, or certain protected species sighting. So it could be a whale, it could be a dolphin, turtle. Then when I talk about a sighting, that is an individual IR camera detection which has been confirmed again by 2 independent analysts as being either likely or certain, and it's a whale sighting. And then there's also some additional human-denied detections that were spotted by the camera but that are not presented in here. So that's not part of the data that I'm presenting. So looking into the results, the first thing that we looked at was the distance of the sightings. So we can compare here the PSO performance versus the IR camera. The IR camera didn't see anything kind of up close to the vessel, and that's because of the configuration. So you can see in the little diagram below that that's really just a function of where the camera is installed and where it can see. So that also correlated with kind of the short range. In any case, even if you see the protected species, typically you can't really do too much about it by the time that you spot the species. You don't have time to react and kind of alter the course of the vessel. So the camera really is not really seeing anything at those short distances, but we can see that it was quite effective at longer distances, so the bulk of it being from kind of 1 to 5 kilometers was the best range for the camera, so those sightings were really, there was quite a few of them in comparison to PSO sightings, so we can see that that data was quite conclusive there. The next thing that we looked at was wind conditions to see how the camera performed with maybe stronger winds. You can see here the data as a function of Beaufort conditions, so from 1 to 7. The red bar is the percent of the time that those conditions occurred. So for example, for Beaufort 2 conditions, that took place 25% of the time over the data that we had. So we can see that the data for both PSOs and the IR camera tracks relatively well with kind of the percent occurrence, assuming that the whales are surfacing no more, no less at different wind conditions. So we expect it to be relatively consistent with the percent occurrence, which is relatively the case. For PSOs, we can see that it's starting to trail off a little bit at Beaufort 4. So as the wind conditions increase, it becomes more difficult, obviously, for PSOs to see the whales. The camera was perhaps a little bit more consistent there with, you can see that it tracks maybe a little bit more closely with the percent of time, with the exception of higher Beaufort conditions where it was probably seeing more waves and confusing the waves with the whales there. So it's kind of an interesting conclusion to see that the IR camera was able to detect the whales in all wind conditions up to Beaufort 7, which was kind of the maximum condition that we had. The next factor that we looked at was atmospheric conditions. So we do have an understanding that thermal imagers and long-wave IR cameras are hindered by damp moisture because the water absorbs the wavelengths of light in the infrared spectrum. So we do understand that if there's heavy fog, that the camera might not be as effective. So we wanted to test that fact and just see what kind of detections we were getting. You can see again the percent occurrence to kind of give us an idea of how long these conditions occurred for, and most of the confirmed detections from the IR camera occurred during the clear to moderately clear atmospheric conditions. We did have some detections in hazier and light to moderate rain conditions, so we thought that that was an interesting finding, and these conditions did not occur for very long duration, as you can see here, so 2 to 3% of the time. So it's a very small sample, so we're not drawing definitive conclusions from here, but we're finding that the camera has detections in low visibility conditions, which is encouraging. And then the last factor that we looked at was the performance of the camera during nighttime. We can see here that the camera had a very high number, relatively, sightings during nighttime versus daytime. So again, you can see the comparison to the percent of occurrence or percent of the daytime. So on a relative basis, we found that more whales were spotted at night than during the daytime. Again, it's a short sample. It's not a very large sample to draw conclusions from, but this is consistent with findings from Zitterbert and al. That said that potentially for them who conducted a similar experiment in something akin to this in Antarctica, they found that there was also more sightings at night, and they thought that this might be due to the lack of glare and diffuse reflections. We thought this could be the case here, could also be due to wider thermal differences. The air is cooler at night, so that delta temperature is greater, so that could be helping the detection as well. In any case, the finding here is that the camera performed very well at night, especially in comparison to PSOs, for who it's very difficult to see at a long distance, so particularly the whales at night. They were seeing bull riding dolphins at night, but they were not seeing any whales. even though they were deployed and they had night vision equipment. So the camera could be a very helpful tool for detection at night. So overall, the camera had an excellent detection rate, at least for our expectations. Of all the PSO sightings, there was only one whale that was not auto-detected by the camera. You can see here the circumstances, the Beaufort conditions and all of that. We don't know exactly why that whale was missed, but it corresponds to a percent detection of 90%, which is in line with other numbers we're seeing in the literature. So we're finding that this was a good result. The detection rate in terms of the number of confirmed blows per hour in the field of view of camera, we had 0.086 blows per hour for the IR camera and 0.026 blows per hour for the PSOs. So in terms of takeaways, there were over 600 detections by the IR system, with over 500 of those classified as only possible sightings, which of course leads us to say that the system needs someone to look at it. There's too many detections, and this is not a replacement for the PSO, definitely not in the foreseeable future, for us at least. We're thinking of this as an additional tool to help the PSOs, potentially also in more difficult conditions. Glare is also one of them. It's difficult for the PSO sometimes to see in glare conditions. The camera can be a great tool. The camera doesn't get tired. The camera doesn't get distracted. So we're seeing this as a powerful tool to help us protect marine mammals and other protected species. Another key takeaway is that, of course, we did not see that many whales in the Empire Wind project area during this survey period, so we acknowledge that this is a small sample size, and it would be very interesting to conduct broader trials on this technology to really gather more data and have more conclusive results. Some sightings by the IR system in low visibility conditions were possible. We'd love to explore that further and try and understand the limitations of the system and what benefits we can get from the IR camera in low visibility conditions, maybe expand when we can do work or protect the species. And then finally, nighttime detection rate by the IR camera was quite high, potentially even exceeding that of daytime detection rates, which again makes us think that this could be helpful at night to protect marine mammals. Thank you very much.
Thank you so much, Audrey. So now we're going to welcome our final speaker for this session. Take a look at the questions that have been submitted and add your upvotes. And now let's welcome Carmen Bernett, who's going to talk about eDNA.
Let me just get myself set up here real quick. All right, well, good almost lunchtime, everybody. Thanks for sticking it out to the end of the conference with us. I'm Carmen Bernett. I'm Deputy Director of Environmental Compliance and Strategy for Invenergy's Leading Light Wind project. While I'm here, I'd also like to acknowledge members of my eDNA team. That's Angela Martinez-Quintana, Darren Kriegel, and Jacob Fredrickson with Fugro, and then Stephen Hill, Beverly McLanahan, and Alistair Wallace with eDNA Tech's Center for Environmental Genomics Application. And just to note that SIGA is one of the few ISO-certified eDNA facilities in the world. You just heard Jason present earlier today on eDNA, and To make it stick, I'll be giving you almost the same presentation, but with just a little bit more information on how we're applying it to the Invenergy project. So overall, my goal today is to impart to you a better understanding of what eDNA is. So you should be experts or dangerous enough by the time you leave today, and understand how Invenergy's project is exploring it as a tool for environmental impacts, mitigation, and monitoring. So just to orient you on the project, we're one of 6 offshore wind leases in the New York Bight, awarded in 2022. We're that little blue polygon down there. We're 40 miles off of the coast of New Jersey and 80 miles from where you sit here today. We're currently in the process of developing our construction and operations plan, and that plan requires us to do a comprehensive suite of studies on the project's impact to the environment. 2 of the resources we're looking deeply at are marine and benthic environments, and we'll go on to see how we apply DNA to that. So just as a primer, DNA studies are not new. They've been going on since 1800, super slow at first and accelerated exponentially since the 90s. For instance, the DNA sequencing techniques I used for my dissertation almost a decade ago has already been surpassed by new technology. And I'm like talking to, the ones that are more up on it this conference, and they're like, oh, that's old stuff. So what I'm talking about today is pretty amazing. So just a primer again, we all know that all things have DNA, with some few weird exceptions that we're not going to go over today. And as a result, living things leave traces everywhere: you, me, fish, sharks, whales. For marine organisms, that's released by way of cell shedding and excretion. And then the application of eDNA analysis is done to what's captured in the environment. So just to lay it down so you really understand it, it's different from what you know in popular culture. It's not 23andMe. We're not studying ancestry. It's not like your favorite serial killer podcast or that CSI show where they're collecting DNA and trying to identify individual, who that killer was. Here we're looking at community composition and biodiversity. We're trying to understand questions like, in this big mix of DNA soup, from different organisms, what species are present, are target species that we're specifically interested in the sampled environment, and what does, the species distribution look like, and, get some inference on abundance. For the Leading Light Wind project, we determined that eDNA can be a really useful tool to assess biodiversity in both water and the seafloor substrate. That can be especially useful if we want to track community composition over time, like pre-construction, post-construction operations. We can use eDNA to track how things are changing over time. Our project is currently conducting boat-based surveys, which are required by regulators to do all the numerous studies that we have to do for our COP. And we decided to take advantage of 2 of our surveys that were deployed in fall 23 and then more recently in spring 24. To test the application of eDNA. And we really think that incorporating this technique is going to help us in the future as we develop our mitigation and monitoring efforts for the Leading Light Wind project specifically, but for all our future offshore wind projects. Did it go through? Okay, here. So this study was only conducted in our lease area. Pretty small sample in comparison to other efforts of eDNA generally, but we identified 17 sampling stations, which we collected a liter of water, like Jason showed earlier. We collected 2 meters above the seafloor, and then we collected also 5-gram sediment grabs using Fugro's specialized grab apparatus. And then standard protocol, you take those samples, you filter them, you freeze them, and then we resulted in 73 water samples and 65 sediment samples collected just for the fall, and that's what I'll be talking about today. I think Jason covered most of this, but just to hammer it home, you collected the DNA like I mentioned. Those samples contain a mix of all the DNA in the environment, in that liter of water or that 5-gram sample. And our project in particular is interested in benthic organisms, fish, and marine mammals. So we extracted the DNA from those samples, targeting just those types of organisms. And for my friendly nerds out there that are interested, we used 18S primers, COI primer set, and 12S primers. So the amplification process creates multiple copies of your DNA, because what comes in that 1 liter sample, no machine can read that right away. It needs to amplify it so that it can detect what was in there. Once that's done, then it can sequence and analyze what those potential organisms are. So the outcome is a whole bunch of sequences, and then that's compared to a database that matches them up and tells you what you found. In our case, we use metabarcoding. Since it's a high throughput system, you're able to put a bunch of samples in the system all at once, and it's able to output very quickly. This information was then used to interpret and inform presence and absence, abundance, as well as provide us with a snapshot of the biodiversity and community composition. So sadly, I don't have a lot to share by data because I just got this last Friday. But all I have for you today are some very high-level findings of what we found in our Elise area. So at first glance, the results yielded a median of about 500,000 sequences per sample. All that means is that's a really good indication that we covered a pretty comprehensive amount of organisms present in the sample. We were able to identify 188 families, 22 phyla, 179 genera, and 98 species. And Annelida, your segmented worms, had the highest number of families at 30, and then the Cnidaria, your jellies, and anemones with 26 families. We also found mid to medium high occurrences in the samples, so about 50 to 75% of the samples contained sand dollar, clams, copepods, polychaetes, and mollusks. And then as for fish, we identified the highest frequency occurring for your stenotymous species or your scups and porgies. They occurred in over 20% of the DNA samples, and then your northern sea robin, there's my arrow, northern sea robin in nearly 20% of the samples. We also detected low frequencies of 2 marine mammals, just some common dolphins, your short-beaked, and then your common bottlenose. I do have data coming in probably this week or next week that's going to tell me a little bit more about what commercial species were present in the lease area and if there were any protected species present as well. So the next logical question is, so what, right? On its own, eDNA results may not be impressive enough for some, but combined with other data that we are collecting and developing, like you've got sediment characterization, visual presence of invertebrates. We are taking videos of the seafloor, views of the seafloor. We've got our geophys ground model coming in. We're doing an essential fish habitat analysis. We have PSOs on all the vessels that we're sending out, and we're collecting marine mammal data and protected species data for those. And then we're also conducting fisheries analysis. Combine that plus your public data and any partnerships that we might decide to do in the future. I think we'll be able to tell a really fulsome story about what our lease is telling us, what's there, and how we can coexist with it as we develop our project from pre, post operations and decommissioning. Oh, sorry, I didn't do the animation. You'll have to sit through it. All right, there you go. So we feel like our efforts were very successful to incorporate eDNA in our survey. We were able to obtain data that can be more specifically used to more specifically qualify the benthic and marine environment, especially for the species that we're interested in tracking. We've proved that this technique is really non-invasive, non-destructive, only required 1 liter of water, 5 grams of sediment for each location, and we were able to qualify the presence of organisms at all trophic levels. And for developers, we were also able to save costs by using already planned surveys to conduct multiple sampling efforts. And I think that'll be especially useful once we start deploying vessels for construction, and operations. We'll just piggyback on those and not have to pay for big vessels to go out and do, very costly surveys. Like any technique, eDNA has its limitations. These can be controlled for through quality control. Your database is only as good as the information that you put in there. So I'm hoping, as this exponentially grows, we're getting better primers, better matches, being able to capture those rare species that we're not able to capture, like Jason had showed in his presentation. So at present, like I mentioned, trawl surveys are used pretty frequently for monitoring offshore wind projects, and we all know that that's it disturbs the environment a whole lot, and so it can result in a lot of bycatch of non-target, juvenile species, protected species, and overall causing death, and we want to limit that as much as possible. So I think with eDNA technology so readily available, the challenge is the time is ripe, right, to step up now and use this. We can't wait for perfect techniques to get us there. So we can assess, I think, what questions we're trying to answer with trawling and see if those questions can be answered by non-extractive methods, or at the very least right now, a combination of lessening those extraction methods and increasing non-extractive methods. So I'm not preaching that eDNA is the be-all, end-all. It is growing pretty fast, and maybe in 10 years, it might be. But perhaps there's a way now that we can consolidate, approaches so that we're able to commit least harm, still get the answers that we seek, and incur the most benefit biologically, regulatorily, and commercially. So thanks so much for sticking around with me and the group that presented today. I hope you learned something new about eDNA, and I hope you're able to get lunch and get home safely and enjoy the weekend.
Thank you so much, Carmen, and thank you for being gracious about me mispronouncing your last name, which is not Bennett, it is Carmen Bernett. Thank you. So here we are ready for the Q&A session. I'd like to invite our speakers to come back up, please. And I would like to invite anybody who's sitting to stand up, do a little stretch, move around a tiny bit. We're really getting there. If you would like to add any questions, I think you can still do that. If your question doesn't get answered, then there is contact information for every one of our speakers on the app. So you can contact them directly or perhaps try to catch them after we're wrapping up today. So I'd like to start with a question that came in from Tricia, and this is for all of our speakers. Can you talk about data management for these technologies? How are your data stored? And are there currently data standards for how they're collected, archived, or analyzed?
I can go first on that. I'm very fortunate to have the great Rutgers glider team who have built a great glider DAC, as well as there's a glider DAP hosted by NOAA, so a lot of the glider data in terms of the physical oceanography data and the optical data and the data collected by the glider itself is very standardized and publicly available in most cases. I will say the acoustic data that we're collecting on the whales is not on those services yet, but I know that is an active project on the Rutgers side to find a way to make all of that data publicly available.
Coming from USGS2, so I mentioned Black Pearl, where we archive all of our imagery data. So that links really well to Sciencebase. I'm not sure if others have heard of that. That's how we publish our imagery data sets and all of our other data sets. So we'll publish like everything we collect through Sciencebase, make it accessible to the public. That's the imagery side. For the data side, so as we collect annotations that the machine learning models need, there are plenty of standards that we follow for, commonly formatted machine learning data sets like COCO or Pascal-Vac, things that are commonly used in the community. So that's what we'll be releasing our data as well.
I didn't have data, so I'm going to pass this along.
For the IR camera, the data is not currently being made available. I suppose it's something that we could think about. The only thing is that there's quite a bit of QA/QC that needs to be done, and it's very it requires a lot of processing. So as I mentioned, that we had these two analysts that were looking at the data and interpreting the data, and these people need to be trained. So I think there's a bit of a barrier to just sharing the data given the analysis that's required, but it's certainly something that we can think about.
I mentioned we had several analyses ongoing, so not everything would be available publicly just yet, but the eDNA data we're definitely going to be sharing publicly. We just have to figure out, what platform and how to do that after we QA/QC the information. And also, just waiting for the results for the spring data. So we'd be happy to share that.
I think that's a great question, the data management question. We developed a sample tracking and database internally just to allow us to keep track of stuff. And our issue was eDNA, environmental, and trawl data sets coming from like 3 different sources and make it all talk to each other and sing together. It's been a lifesaver developing that. And our data is also publicly available. It's not posted anywhere yet, but it's state-funded data, so it's publicly available. But I didn't appreciate how important that was to what we're doing until I started to lose sleep at night over what's what.
Okay, thank you so much for those responses. There are many questions about eDNA, lots and lots of excitement about eDNA, which I share. And some of those questions are about, do you know how far eDNA can travel in the water? Do you know about the rate of degeneration? And what is your confidence that eDNA samples are representing the community where you're collecting your sample from?
That's a great question. I kick myself for not. That's usually part of my what is eDNA. We're fortunate it's short-lived in the environment. There's 2 main things that make the signal go away after it's shed: physical dispersion and movement. And we've partnered with RU Cool and students, Josh Kohat and I, and Hugh Rorty, co-mentored students using their surface current models to model that. And biological degradation, because nucleic acids are great microbial food. We're fortunate that it goes away quickly, because when you detect DNA, it's something that was shed recently. It's not last month's signal. It's not even last week's signal. The half-life of eDNA in aquatic environments is 1 day. That's the going rate. There's variability around that. So given that, the confidence I have that what we're measuring comes from where we're measuring comes mostly from the artificial reef stuff that I showed. I was really surprised that within that small area, which is on this order of kilometers, a few kilometers, that we resolved different communities that corresponded with their trap catcher on the artificial reefs capture survey, and different from the surrounding water column away from the reef. So I think what we have to remember is, although eDNA is advecting in from other areas, it's being measured on top of the local signal at the time you take the sample, which is going to dominate the signal. And the techniques that are used are sensitive to the more dominant genes in the PCR reaction, for instance, when you do the amplification.
I think you got it.
Hey, curious, how fast does it sink?
Yeah, I want to point to Josh. We haven't gotten to the vertical advection yet. So it comes sometimes in cells, sometimes it's in chunks of tissue, sometimes it's in a cell. So it's like the phytoplankton story. It depends on the size and the shape and how it's packaged in the ocean, how it's going to vertically move.
Could you hear that at the back? Great. Okay, so let's give the eDNA folks a little rest, and we'll certainly come back to you. But I want to hear more about whales. So, Katie, have you been able to look for linkages between your detections and other maritime activities? And what kind of, if you have, what kind of data have been most useful for that?
So not really yet. The focus, because of the NYSERDA funding, has been on the wind lease areas, but obviously the gliders are transiting through shipping lanes. We prefer not to keep the gliders in shipping lanes longer than we have to, just because there is a collision risk, especially when they're doing that surfacing behavior. They'll sit on the surface for about 20, possibly even 30 minutes transiting data. So we don't want them to get hit by boats, so we try to minimize the time in congested areas. But it is definitely something that I could look at in the future and just haven't gotten to yet, so that's a great idea. Thank you for whoever asked that.
And if anybody who's here has suggestions for Katie, come meet up with her after the talk or find her contact information.
I'm from Stony Brook. I have nowhere to be after we're done today.
Thank you. Kyle, that was some pretty exciting artificial intelligence research you were showing us. I think you gave us all a lot of hope that this technology is going to be used for good, or at least not just for some scary things. Could your model be applied to other species, things like large forage fish schools? And do you know whether there have been any studies to look at the accuracy, precision of these detections?
I'm not sure about, I'm not aware of any studies, although I haven't looked too hard specifically into schooling fish. But we had an interest to detect individual objects, so like a bony fish within that broader fish shoal. You could easily aggregate that up to say if bony fish are particularly dense, that becomes a fish shoal, and then you could train a model to make those detections happen. The data actually came to us as identified fish shoals, and we broke that up into more fine, like individual objects. The unfortunate annotator that had to do that, I'm apologizing for. It was a very tedious process. Yeah, good question though.
Yeah, thank you, Kyle. Now we have a question for Sarah. Does data transfer remain an issue if something like Starlink is available? Is the size of data too much for a satellite link?
That's a good question. It really depends on the data. And when you're doing monitoring too, it depends on whether you need a real-time answer or if you're archiving data and pulling it in later and doing an analysis after the fact. Some types of data are really big, like acoustic data, for example, is difficult. What we usually have done historically, and maybe AI is going to be a good help for some of this, is rather than transferring all of the data by a satellite, you have something in situ that sort of detects where there's something interesting and just clips the data and sends it. So if you wanted to look and see, oh, do I have a whale call here? You could do that. So some of those data transfer systems are really, we can certainly use satellites. But one of the benefits of having all the cables out there is, I'm hoping that we'll be able to find some ways to also integrate data transfer through cables so that we can move larger data sets and maintain more data rather than clipping all of it to like smaller pieces. But some of that it also involves cybersecurity risks and other types of issues that we need to work with developers and manufacturers of cable and stuff to make sure that if you want something in real time, it's probably not the best solution. But if you're going to archive something and do an analysis later, it may be really good for moving very large data sets.
Great, thank you, Sarah. large data sets is going to be a common theme, I think, in the years to come, right? So Audrey, we got a bunch of questions about infrared whale detections. I think people are pretty excited about having a new technology to protect whales and to also help the projects be a little more efficient in their construction, I think. So one of the questions was there a stabilizer on the infrared camera? And another was, are those camera detections reported in real time on the vessel? And I would add to that, are they reported outside of your vessel to any other organizations?
So yes, there is image stabilization. It's electronic image stabilization. And the second question was if there is real-time data action. So right now, we're not currently using real-time information, for example, for the pilot or anything like that, because of the fact that we're just in trial mode. But it's something that is kind of a low-hanging fruit, and that is on the radar to eventually put some live positioning of where the whales are, and perhaps allow the vessels or PSOs to take action a little bit faster based on the information. We could also potentially think about putting the information into a tool like Mysticetus that would give other people, I believe, information on that. So I think that that's something else that we could be looking at in the future.
Thank you. So I have another eDNA question for Carmen and Jason. Do you know if there are eDNA artifacts from ingesting prey and subsequent egestion of waste? In other words, do you detect prey eDNA from a predator's excrement or fecal matters? And do you know if maybe that methodology is being used on any other species, getting DNA from feces to look at diet?
So my research in grad school was onshore and on land, and I had a colleague who was using scat and eDNA collected in scat to track coyote movements. So yes, it's possible to use that. I don't know, Jason, I'd defer to you on how long it hangs out in fish poop. But I mean, in coyotes it worked. She was able to map out their range because it was just easier to find scat than it was to find the coyotes themselves.
The only application I know of like that for fish contents, people use metabar coding, and Jackie Sullivan, who was here, uses it to look at fish DNA in, is it harbor seal feces? So-called fDNA, fecal DNA, yeah. But I think the first part of your question was, if something eats something over here and defecates over here, will we detect its genes? Yeah. I mean, the primers aren't going to differentiate. If it's the genes there, it's going to be detected. The main thing we need to be aware of is that we're not sampling fish. We're sampling fish genes, which you made the point very well, Carmen, in your presentation. It's a different, you have to have a different perspective.
You know, a quick thing about that, I mean, I actually did DNA work as my PhD too, coincidentally. And people were using that in my lab. Yeah, people use that technique in my lab too, but the DNA was more degraded in the fecal matter. So I think that there is the feasibility to be able to maybe, based on like quantity and degradation, to understand the difference if we, wanted to get more into the weeds on something like that.
Thanks so much. I'm going to send this question to Katie. Can you clarify points about year-round and year-round detections in every or most month? We have made some clear distinctions in our presentations and papers based on detections per day over months throughout the year.
Yeah, I could definitely touch on that. So the detection rates that we're getting, which I'm quantifying as the proportion of days in a month that we first had a glider in the water, that is the big caveat. We don't have a glider in the water every day of the year, especially in June for some reason. But of those days in a month, what portion of those had a whale detected? And I'm detecting the whales on daily presence. Like I mentioned, I'm using Mark Baumgartner's low-frequency detection classification system and call libraries produced by Genevieve Davis. And I'm also using her thresholds for daily presence, which are based off of slightly different hydrophones, as well as stationary moored platforms. So I'm still kind of, I think those need to be tweaked a little bit for gliders, especially for the fin whales. So there is some fine tweaking that needs to get done in terms of the daily presence absence proportions that I'm reporting now. I had another thing I was going to say and I lost it completely. So hopefully that answers your question.
Yeah, thank you, Katie. So we have a question for our 3 eDNA folks up here now. Can you tell us a bit more about kind of detections on a very broad spectrum of taxonomic groups? Like, can we get the lower trophic levels? Are they coming through at reads that could be detected? I probably used that word wrong.
No, I think that was right. You had different primer sets. I'll let you start.
Yeah, so the way you detect in a community is based on the primers that you use. So we were interested in the benthic environment, so we used primers that would detect benthic organisms. We also wanted to learn about fish, so we used primers that would detect fish. And then the same for the marine mammals. So like I said earlier, your detections are only as good as the primers that you use. So if you have a specific organism that you're looking for, but you don't have the primer for it, it's not going to turn up in any of your eDNA samples, even if it is there. So it's just a matter of how you develop those primers. Some places have specialized primers that they develop, and then there's some that are publicly available, that are published in papers that you can use, and you can have them built to satisfy what you're looking for in the environment.
I'll just add to that I think it's hard to appreciate how amazing an environmental DNA extract is. The amount of information in the molecules in that little, nanograms of DNA that you get. And as you're saying, you can choose which section to look at by the primers you choose. So that's a benefit, but the challenge is if you want to do bacterial, that's 16S, phytoplankton, there's going to be some 16S and like 18S and 23S, and then vertebrates, there's 12S. There's different primers for fungi and invertebrates. I think you used 18S or something. 18 COI. So each of those primers is another sequencing run, which is just multiply the cost of your sample by how many primer sets you use. And that's the challenge. So it's all there, but it's not cheap to access.
Yeah, I mean, I think it's sort of like, when you get a blood test and they don't check everything, like they're looking for something and they put, they do the test associated with what they're looking for. But you can get to, I think what you were getting at too, is like you can get to different levels of, like, whether you're looking for a species versus a phylum or something. Yeah, you kind of have to decide what is important, and then you get down to that level of specificity with the primers. And you can develop primers. If you have something you want to look for specifically, you could actually go about making a primer that does that. So I think in the future, like you were talking about, 10 years from now, you could imagine a library of all kinds of different primers for this purpose.
One more quick thing. When you get your DNA extract, it's typically 100 microliter volume of extract, and about 5 microliters of that is used in the amplification reaction for a given primer set. So on one sample from 1 liter of water, you have the capacity for doing multiple primers.
Thank you so much. So we're down to just a couple of minutes, and we probably can't answer this question in that amount of time, but if you would like to, by all means, what's next for your research? What do you think is the most necessary next step for the work that you're doing to be really valuable to the questions we all have about offshore wind and wildlife and fisheries?
That is such a loaded question. Gosh. I think we really, in terms of gliders and acoustic, passive acoustic monitoring, I think one of the sort of big open questions, at least for me, is can you use fleets of gliders to triangulate whales? Which is something that's very difficult to do, even in a moored situation. But I think with directional hydrophones and fleets of gliders, that'll also help you tease out variability in both space and time and all that kind of fun stuff. I could go down this rabbit hole. Josh Kohut and I have had this conversation several times. I will spare you all the details. But I think that sort of a fleet-based or just continuous monitoring to look at the interannual variability is really where we need to go to get a good idea of just how whales are using this space and then to build a baseline on how that's going to change in the future.
Great, thank you. Can you give us a bullet, Kyle? Sorry, we are running out of time.
Yeah, AI is very data hungry, so what's next is we need to collect more data, like more species, more examples of current species we have, more geographical areas. We're expanding our flights to the Gulf of Mexico, so my short bullet.
Thank you.
I would say I'm lucky enough that people are actually doing the next step now. Josh is sitting out there, you know, the Rutgers folks and their partners and team members are using, you know, the information that we've brought together to help inform work they're doing on creating more holistic monitoring approaches and determining what can be monitored. And then I see Julia back there too. Now RDC and RWSC and other folks are considering those outcomes in terms of how to structure calls for funding, you know, calls for proposals on technology. So it's been really actually very satisfying to see that work moving forward with a lot of other folks and after we finished our project.
So I think for the toy on camera, we've already discussed a few things and kind of improvements that we can make, but the next big step for us will be to actually utilize the camera on our Empire Wind project when we put the foundations in, which should be next summer. So we will be deploying the cameras then and hopefully gathering a lot of data. So I think that will be the next step for us to really prove and think about what we can do with this technology.
Near-term next steps is we've got our spring data coming in, so we're going to compare that to our fall data and see how things shake up there. And then considering how, you know, all the data resources that we're pulling together will be corroborated against the eDNA data that we're going to be getting results for. And then once we have that, we'll probably try to apply it to our even keel wind project in the California coast. And that would be interesting to see what kind of critters we find on that side of the ocean.
Monthly or lower frequency shore-to-shelf transects of eDNA sampled by AUVs and lunch.
Okay. Let's give a nice warm thank you to our speakers today. Thank you.
I'm briefly going to do one more round of thank yous because this week has been excellent. It has gone really smoothly. First, I want to thank the folks at Stony Brook because they've been wonderful. Thank you to all the AV folks and everyone else who has been helping us move through these rooms, feed us, and everything. I would like to thank again the folks on our planning committee. So our NYSERDA events team has been fabulous. Also, all of the people from Biodiversity Research Institute and Tetra Tech who have helped us keep time, manage questions, in addition to their roles on the scientific planning committee. I would like to make sure that we thank everyone who helped organize the sessions. This was an amazing amount of information this week, and the session organizers put together fantastic panels and presentations. Thank you to the moderators who managed so many more questions than we anticipated, and we will try to make sure that we continue to address some of the questions that have come up. Thank you to everyone who organized a side meeting or workshop. Those were fantastic yesterday, and just a real benefit, I think, of bringing us all together is the opportunity to kind of push on some of those issues and make some more advancements. And then, we already did that. Yeah. I guess lastly, I need to thank Kate Williams and our scientific planning committee, and Kate's going to go over a few of the other follow-up things from this workshop, but I cannot say thank you enough to Kate for really spearheading the scientific planning of the workshop and putting together a fantastic agenda for all of us this week.
Thanks, and since Kate couldn't thank herself, thank you also to Kate Press for putting a ton of work into making this happen. So we have a few quick things.